All right, welcome to our lecture on mutation. So just before we get started, excuse me, Corona's killing me. Before we get started, I want to point this out. So as we go through the lecture today is kind of like it's a hands-on sort of activity. So it's not just a lecture, you take notes. That's part of it. Like we're still going to be using the note sheet as we go through this. But the part that I want you to pay attention, the part that I really want you to work on is when I say pause the video and then do the translation yourself. I really mean that. Like pause the video and then like you see if you can do the translation, like you can turn the sequence of nucleotides into amino acids. And the reason why I want you guys to do that is because on the exam, I'm going to have questions where I show you the original strand of DNA and then I show you the mutation and then I ask you what type of mutation it was. And you're not going to be able to know unless you actually go through this process. So please pause the lecture when I say pause it and see if you can figure these out. Okay. All right. So let's get started. What is a mutation? So mutations are changes in the DNA sequence, but at the level of the nucleotide. And so that means like if your nucleotide sequence reads like ACTG, that would be a mutation where now it reads ATTG or something. So it's where the nucleotide sequence changes. And since the nucleotide sequence parallels this sequence of amino acids in a protein, that a mutation could affect the amino acid and then the overall structure of the protein produced. So let me give you an example here. So let's just take a small strand of DNA. Okay. Doesn't matter which side is a template. Doesn't matter which side is the anti-sense or sense. None of that matters, right? But what I want to show you is that if there's a change in the nucleotide sequence, let me grab my pointer. So in this particular case, this base pair, C and G, were changed to the base pair A and T. And so where anytime that you have a substitution where one nucleotide is substituted for another nucleotide, it's a base pair substitution. So that's where one single nucleotide pair, like we just saw on the previous page, is substituted for another. Okay. The other type of mutation is where a nucleotide is actually inserted, like an extra nucleotide comes in, and then the other type is where it could also be deleted. And so we kind of pair those together because they have a similar effect on the amino acid produced. Okay. So let me adjust my mic here. Okay. Where was I? So let me give you an example of both of those. Mic's all being weird. Okay. So let me give you an example of both of those. So we have a gene here, right? It doesn't matter which side is template or sense. And you can see the base pair C and G. And in a single nucleotide, a single base pair substitution would be something like where that G and C gets switched to an A and T, right? And so it doesn't change the length of the gene. It doesn't change the length of so it doesn't change the overall length of the gene. So like the number of nucleotides hasn't changed, but the order and sequence has. So instead of it going ATCG, it goes ATCA. And so that might affect the protein produced. Now, the one where it's the insertion or deletion, in this particular case, right, so it goes ATCG, and then look, the G actually gets removed. So that would be a deletion, and that does change the length of the gene. It does change, it does change the length of the nucleotides. And then here's one that goes ATCG, and then we go ATCAG. So we added one in there, so that's an insertion. And so now those are the two types. Those are the main two types of mutations. Now there's a ton of other types of mutation that they're like bigger mutations in a way where you can take an entire segment of DNA, duplicate it, and insert it somewhere else in the genome. And that's actually happened a lot, like in the history. We'll talk about that a little bit when we get into Unit 4. They're called transposons, and the idea here is that a long time ago, like way before people and mammals, but back in the times like when the seas were swimming with early life, nature was experimenting with different body plans. And so what they would do is nature would duplicate this region of DNA that coded for how to make like a body part, and it would duplicate that segment of DNA. And then that body part would get duplicated. And so you see this happened a lot with like millipedes and centipedes. they ended up with these mutations where they end up with lots of body segments, and so that's a different type of mutation. But I digress. Okay, so the main two types though that we're interested in here are the single base pair substitution, a point mutation, or the insertion and deletions. Okay, so we can say, okay, what is the effect of a point mutation? And there's going to be many, right? There's many possible effects. So one effect is a silent mutation. So that's where like there's a single base pair substitution or a point mutation. So a single nucleotide is changed, but the actual amino acid isn't changed at all. And so we can see an example of this, right? So what I want you to do, okay, pause the video for a second in just a second. So what I want you to do is imagine the top strand is the template strand, okay, and the bottom strand is the sense strand. So remember that when the mRNA is produced, it's going to look just like the sense strand. It's going to read exactly the same, except the T's will be U's. And then you're going to take that mRNA transcript. So you're going to have to make it right now. So you're going to pause the video and make the mRNA transcript. And then you're going to come over to our codon chart and you're going to translate the codons into amino acids. And what I'd like you to do is use the 3-letter abbreviations. So if your first codon was A, U, G, so first position A, second U, third G, your amino acid would be methionine. So you could draw a circle and then inside the circle write MET, and that would be the first amino acid on your chain. All right, so go ahead and pause it and then translate that. All right, so if you pause it and you translated that, what you should have is AUG is methionine, CGG would be your next one. That'd be arginine, so it should say ARG, right? The next one would be CAG, so CAG would be glycine, GLN. The next one would be CCC, so CCC would be proline, PRO. UGU, so we come up here, U first position, G second position, U third position would be cysteine, CYS, and then the last one, UGA, that's our stop codon. Doesn't code for an amino acid, it just has that release factor, right? The termination factor. Okay, so now let's imagine there's a mutation, okay? So you're gonna do exactly the same thing we just did. You're gonna take the sense strand and you're gonna convert the T's to U's, and then you'll go every 3 base pairs as a codon, and then come over your codon chart and translate it. Go ahead and pause the video and do that. All right, so if you pause the video and did that, what you found out was the first amino acid was still the same. AUG is still AUG, that's methionine. The second amino acid, CGA, right? CGA would be arginine. Oh, wait a second. That didn't change the amino acid at all, right? CGG was our other one, right? CGG coded for arginine, and so does CGA. So no matter what, even with this mutation, the protein that we make is still going to have arginine as the second amino acid. And so we'd say that was a silent mutation. So there was no effect on the amino acid. Let's try a different one. So we're going to go into missense. Okay, so we got another change in a missense is where when we have that base pair substitution, that substitution leads to a change in the amino acid, just in that one amino acid. All right, so let's go ahead and practice this, right? So let me give you an example, okay? So here's a brand new gene, okay? The top strand is the template or antisense strand. That's the one that RNA polymerase reads and makes a copy of. The bottom strand is the sense strand. And so instead of trying to convert the template strand into like an mRNA transcript, just look at the sense strand. Sense strand, copy it exactly the same, and then just change the T's to U's. So it should read AUG, UCC, UUU, and then you're going to take that over here to this codon chart, and you're going to translate it. So go ahead and do that real quick and just figure out what the sequence of amino acids would be. Okay, so if you did that, you should have the sequence should be AUG, that would be methionine, UCC, so we got UCC, that'd be serine, UUU is going to be phenylalanine, PHE, and then we got ACC, so ACC would be threonine, or THR, and then you'd have GCG, GCG would be alanine, And then we have UAA. UAA codes for stop. Okay. All right. So here comes your base pair substitution. Here comes your point mutation. All right. So go ahead and translate this one. Just see what happens, and let's compare notes. Okay, so hopefully you pause it, did that, and then let's figure it out. So the first one hasn't changed. AUG is still the same, so that's methionine, MET. And then we have this base pair substitution in the second position in the second codon. And so instead of it reading UCC, it now reads UAC. So let's see what happened. So it was UCC, which was serine, right? And now it's UAC, UAC. So now it's tyrosine. So the amino acid was changed. It went from serine to tyrosine. That was a missense mutation. Okay, let's go on to the next one. So the next mutation is one where the nucleotide or the base pair substitution leads to a change, but it's not in the amino acid. It changes the codon to code for a stop codon, like an early stop codon. So what ends up happening with a nonsense mutation is the ribosome will be translating the mRNA. It'll be adding amino acids, and then it'll get to the stop codon early because of the mutation, and then that the termination factor will bind into the ribosomes, a large subunit, separates the two sides, and it stops translating, and then you're stuck with like a shortened polypeptide. So it's an early stop codon. So here's our example, right? So this is all good practice. Go ahead and pause the video and translate this gene. Okay, so if you pause the video, you should have your mRNA transcript should be AUG, ACC, UGC, right? And then what we'll do is we'll translate it. So AUG, always methionine, ACC, ACC, threonine, UGC, UGC, cysteine, where was I? UGC, GCG, GCG, alanine, CGG, CGG, arginine, AAC, AAC, aspartic acid or something. I don't remember what that one is. ASN. Where were we? Oh, did I do that? ACC. Yep. No, did I do that wrong? Son of a bee. Hold on. I don't have mine broken up into spots. So let's do this. So you're probably like, oh no, did I do it wrong? No, I was doing it wrong, so I was doing it in my head. Okay, methionine is the first one. ACC, that one is still threonine. UGC, UGC, cysteine, UGC. C or GCG, GCG, GCG would be alanine, ALA. GAA, there it is, that's where I messed up. GAA would be glutamic acid, GLU. And then CC would be CC should be proline. And then UGA would be our stop. Okay, so that's what yours should read. Then we have a point mutation, and let's figure out the effect of that. So take a second and go ahead and translate that. Pause the video. All right, so if you pause the video and translated that, it should be methionine, AUG, ACC, that's still the same, that's still threonine. And then, then it will be, instead of UGC, it becomes UGA. Well, look at that. If I'm a ribosome and I'm reading UGA, boom, stop. Translation stops right there. So you only get an amino acid, or you get a polypeptide that's only 2 amino acids long instead of the full 5 or whatever we were doing. Okay, so that's a nonsense. So nonsense is when you have a change in nucleotide sequence that leads to a stop codon. Now let's take a look at insertions and deletions. So no matter what, if we take a nucleotide out of the sequence, or we add one to the sequence, it shifts how the ribosome reads the codons. It shifts what we call the codon reading frame. So here's an example, and I'm doing this. These aren't nucleotides. These are just words. The cat ate the rat. This makes a lot of sense because we're like, okay, there's a cat and it ate a rat. Now watch what happens if, notice that each word is 3 letters long. Watch what happens when we add a letter to it. And it shifts how we read the reading frame. So we added a letter to the beginning, and now it's ATH, ECA, TAT. So it's ath, eka, tat, eth, era, t. So this is just nonsensical. Adding the nucleotide shifted the reading frame. So now it's going to change every single codon downstream of where the mutation took place. Now let me give you a different example. So the cat ate the rat, still the words we're using, but now we're going to remove. So we're going to delete a nucleotide. So it shifts the reading frame 1 to the left, and now we're reading heck, atta, tet, her, at. Doesn't sound like the cat ate the rat anymore. So the whole thing is changed. That's why insertions and deletions are for sure the most devastating types of mutations when they happen. So, yeah, they're also, so insertion and deletion is the type of mutation. The effect that it has is it's a frame shift mutation. So let's take a look at what a frame shift mutation can do. So anytime an insertion or deletion shifts the reading frame, it changes all the amino acids downstream. So I'm going to give you another gene. The sent strand is on the bottom. Go ahead and pause the video and translate it. Okay, if you pause the video, then you should have AUG, so that codes for methionine, MET. ACC, ACC is threonine. CGG, CGG is arginine. CUC, 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 I see, we see. CUC, CUC is leucine. CUC, and then UUC. So UUC would be phenylalanine, PHA, or PHE. UUC, and then we have ACC. ACC is threonine, THR. AGA, AGA is arginine, and then UGA is our stop. So that's a perfectly healthy gene. Okay, now what we're going to do is we are going to insert, notice we've inserted a base pair. So an A and a T in the middle there. We even spelled cat. Look at that. All right, so now go ahead and pause the video and translate this again and see what happens to the amino acids produced. Okay, if you pause the video, what you should see is that first amino acid, because it came before the insertion, isn't changed. AUG is still the same, which is good. So the ribosome will start translating. So AUG starts off good, methionine, AUG, methionine. And then the next one, instead of it reading ACC, now it's reading UAC. So instead of threonine, it's UAC, it's tyrosine. Okay, and then the next one is shifted, CCG, CCG, now proline. And then if you keep going, they've all changed. And so that's an example of an insertion and then the downstream effects, the frame shifting effects of that insertion. Okay, now let's take a look at this one. Oops. So we'll go back to this beginning one, right? So we added one, now we're deleting one. So instead of inserting one, now instead of having ATGACCC, it's ATGCCC. So we removed this adenine and thymine base pair. So this would be a deletion. Notice that upstream, the AUG from the deletion, still the same. It's our methionine, AUG. Go ahead and pause the video, finish translating this bottom strand of DNA. And okay, if you pause the video and translated that, you should get methionine. And then the next one is CCC, so proline. So we went from ACC, which was threonine in the original one, to proline. And then GGC is glycine. That's completely different than, where was I? So we had AUG was methionine, ACC was threonine. Then we had CGG. CGG was arginine. CGG was arginine. And on this one, we had AUG methionine, CCC proline, GGC was glycine. And then all of the rest of them were also changed. I'm sure you saw that too. So in summary, we can see that the biggest effect is going to be from an insertion and deletion. The biggest effect on the polypeptide produced. The chance that that change, that insertion or deletion, had a positive or a beneficial effect, like a made somebody grow wings or now they can fly is like zero. It would never happen. So most mutations, like the bigger they are, the more disastrous they are. All right. So what causes mutation? This is a this is a question that can be answered with many answers. So, mutation causes. What we see in the movies is pretty accurate, although the effects are not. So mutation can be caused by high frequency electromagnetic radiation, like UV light. So anything that's in higher energy than the visible light spectrum can interact with your DNA directly. It passes through cell membranes right to the DNA, and it will cause damage. And the damage it causes is it will break strands of DNA. Now, your cells have repair mechanisms, which is good. They'll come in there and say, oh no, I have a strand of DNA that got separated and broken by this UV light. Let's repair it. And so the cell will do the best job it can repairing it, but occasionally it'll accidentally insert a base pair, or to remove a base pair, like an insertion or deletion, or it might substitute. It might pick the wrong one. And so that's one way that we get mutation. Another way is if you're exposed to a chemical carcinogen. So that could be something that is a mutagen, something like the ooze in Teenage Mutant Ninja Turtles, right? Something that like some chemical that causes the DNA machinery that replicates DNA to not function as well. Something that actually damages the DNA directly. Any of those would work. That causes the repair mechanism to kick in and do its best job repairing, and then it makes mistakes. Right? All right. Another way that we can get mutation is that if the DNA polymerase makes a mistake while it's replicating. So as we know from our DNA replication lecture video, that the DNA polymerase is traveling along the leading and lagging strands, and it's reading a nucleotide and placing one in. Well, occasionally it reads a nucleotide and it puts the wrong one with it, and so they don't pair together very well. And then there's another enzyme that comes along and kind of scans the DNA to make sure that there's no bubbles or anything weird. And if it finds one, it's going to do the best job it can to kind of figure out what happened and cut it out and put the right stuff in. But sometimes it makes a mistake there, and so um, there's another way. So that's in eukaryotes mostly. Prokaryotes, though, in a state of crisis. So when prokaryotes are exposed to like deadly environments, or you put them somewhere that they're not used to, some prokaryotes will actually change what polymerase they're using, and they'll start using a polymerase that on purpose makes more mistakes. It's just not as accurate. It's like a crappy copy machine. And that's the prokaryotes way of rapidly introducing mutation into their genome, so that hopefully one of those mutations helps that individual survive in the new environment better than the other variants, the other copies, or the other mutations. And if that individual can do better and survive in that environment, it's got that mutation now that will then be passed on to all of its offspring. So all of its offspring will be adapted to that new environment. And so that's kind of a cool thing that prokaryotes do. Yeah. And then let's talk about the flexibility of mutation. So it depends on where the mutation happens in a codon, has a lot to do with the overall effect of that mutation. So as we know, a codon has three positions. It's got one position, that's the one in the middle, two position, that's the one in the center, and then three position, that's the outermost concentric circle. So the first position is always going to have the largest change, because look, if I have a G, that means I can make any of these amino acids: valine, alanine, aspartic acid, glutamine, and glycine. I can make any of those amino acids with a G in the first position. But if I have a mutation that changes a G in the first position to, let's say, like an A, I can no longer make any of those amino acids. I can only make the ones downstream from A. So the biggest change in like the mutation that has the largest effect, at least a point mutation, is going to be in that first position. Now, the second position, there may be some redundancy in the second position. So if, let's say, we have a G in our first position and then a C in our second position, I can only make alanine. Now, if I have, let's say I have a G in the first position and a C in the second position, and that C changes to a A, well, then I can only make aspartic acid and glutamine. So these, right, these 3 amino acids will be the ones that are produced from that mutation. Like before the mutation, alanine, after the mutation, aspartic acid and glutamine. So second position will still have an overall effect, right? Um, Sometimes though, that second position will be a silent mutation, but very rarely. Anyways, the third position is one where it's the most flexible. So if we have a G in the first position, an A in the second position, and a C in the third position, that C could change to a U and you're still going to get aspartic acid. If you have a G in the first position, G in the second position, and a G in the third position, as long as it goes G, G something, you're going to get glycine. Now, one of the things that's not pictured here in this image, and this is where I was kind of getting stuck on the second position, is that all of these amino acids are going to have functional groups that are either nonpolar, polar, acidic, or basic. Oftentimes, a mutation in the second position, if there's a mutation in the second position and it changes the amino acid that it changes to is often of this similar type. So it has like a nonpolar functional group, or it will have a polar functional group, or it will have an acidic functional group, or a basic functional group. So even though it changes the amino acid, it still keeps, generally, a mutation in that second position will keep the characteristics of the original amino acid in the new one. Like they'll have shared characteristics. And so the effect of a second position mutation is less devastating than an effect in the first. So it's in order. So first position is the greatest. Second position can change the amino acid and will always change the amino acid, but sometimes it changes it to an amino acid of a similar type. The third position is the one that's the most flexible. So, like GUU, if that's GUC or GUA or GUG, right? If that third position changes, it'll be valine. It'll always be valine. So that's kind of nice. There's a lot of flexibility in the third position. And so that's the lecture on mutation. Hope you enjoyed it.